Okay, a couple of things. Actually, I could not hear many of you. So I'm going to speak loudly and expect you guys to speak louder than me. And second thing, I noticed that there are a lot of women in this room, but I see questions only from the Y chromosome carriers. Can we have some questions from the XX guys, please? Okay. Okay, so, okay, I will start. Deepa introduced some of the basic concepts already. So some of this is slightly repetitious, but at some point I'll take off to something new. Okay, so already you heard about evolutionary forces. Forces I'll put in quotes because I'm a physicist and forces means something else for us. So what, correct thing to say is evolutionary processes. So you've already heard of them. So there's selection, there's mutation. So all of you have some idea about these two processes. Okay, and we'll discuss that in detail as we go. There's one more called gene flow. Unfortunately, Himani was supposed to cover it and she is unable to make it. But what it means is that we are thinking of gene flow between different populations. Okay, so that we will not be able to cover. And selection mutation also can be between different populations, but we'll basically talk about a single population. And then finally, there's something called random genetic drift, which refers to stochasticity in the offspring number. Okay, so I will start my lecture with describing random genetic drift. Okay, so these are the evolutionary forces. And some of you have been asking this question about genotype, phenotype. So, in fact, there are two broad frameworks in which one describes evolutionary dynamics. One is called population genetics, in which one, most of the work is at the genetic level. So what one wants to know is that under the action of these one or more of these evolutionary forces, how does the allele frequency or the genotypic frequency change with time? And then you want to ask, related questions about diversity and so on. But this is the basic object that you want to calculate. Calculate as in the experiment, in your simulations, in your model, maybe you want to work out some things. So that's one class, one framework, one theory. Another is called quantitative genetics, which deals with mainly phenotype. Okay, so I like to think of genotype, this population genetic is a microscopic theory. So we'll be looking at this DNA and stuff like that. Whereas quantitative genetics is more like macroscopic theory, in which you're looking at some things which is sort of, like height, etc. But the underlying things, underlying stuff is microscopic. So you don't typically worry about that. Okay, so that's about phenotype. And here we're interested in asking questions about variance, which is a macroscopic quantity, and so on. Okay, so we'll see how that works out in perhaps lecture 3. So now you would think that, selection acts on a phenotype, but stuff which is hereditary is a genetic one, right? So why two theories? So there are historical reasons and stuff like that, which I will not go into. But what we would like is a framework in which you combine the two of them. That means I start at the phenotypic level, do a map. So some of you were asking how phenotype and genotype is connected. That's through a map. And I'll come to that later on. So you start with the phenotypic level, somehow connect to genotype, do your population genetics there, and go back and then answer questions about phenotype. So that's a sort of very broad framework, very broad, roadmap in which we can connect these two, microscopic and the macroscopic level. So we'll come to that later on as we go. So what I want to do in the first 2 lectures is focus only on population genetics part. That means we will deal with things only at the genetic level. And these are very, simple-ish questions. So therefore, we'll spend some time, a couple of lectures on that. Okay. So we'll do that. And then I want to give you some references. There are several excellent books, but you can choose whichever one you like. These are my favorite ones. There's a book by Barton et al. Called Evolution. It's one by Charlesworth and Charlesworth. And more recent one is Walsh and Lynch. So you can look up any of these or whatever you like, whichever you like. These three books have both maths as well as a lot of biology. So some of you may be more comfortable with them, with these. But for the more mathematically inclined, I recommend the book by Evans. Hard to read when you're starting off, but if that's where your interest is, maybe you want to look at this. There are also some scientists, some people who work in the area who have their notes, lecture notes put up on the websites. Again, you can find them and see if they're interesting to you. And one very important resource are the ICTS lectures. So like I said in the morning, we have these lectures since 2014, every other year. So there are several of them, very high quality ones. And we will be able to cover we'll be just barely able to scratch the surface. But what else is new? What is going on? What are the interesting questions? We will just stay a little bit only in this week. But please, I strongly encourage you to look at this repository. It's very helpful. Okay. So therefore, you can look at these references and so on. Okay. So I'm a theoretician. I do modeling. And you might worry, why model? So as we go along, I'll explain what good they are for. Okay. So like I said, I will start with talking about random genetic drift. Before we come to that, I find it easy to not think of evolution for a second. I will tell you a very basic model, which actually dates to 1600s. And if I get my, dates right. Yeah. So I will start with something right now. It's not connected to evolution, but I hope it will give you the basic concept. Okay, so it's called a gambler's ruin problem. So it has appeared when the probability theory was just starting, which was in the 1600s. So people were, the origin of probability is in gambling. Okay, people want to make more money. And this was some correspondence between these two physicists / mathematicians, Pascal and Fermat. Fermat, I'm sure all of you heard, so I'm sure Pascal also. So this is about 1600s. Okay. Okay. So the problem is very simple to state. So let us say all of us have some amount of money. Let's say 40 rupees. Each of us go to a casino. Okay. And the rule in this game that we were going to play in the casino is that we toss a fair coin. That means the head and tail are equally likely to come. So if head comes, you win 1 rupee. I win also 1 rupee. So you toss your coin, she'll toss her coin. All of us will do independently. Okay. So if you get a head, we get 1 rupee. Okay. So these are the rules. So we have a toss a fair coin. Head gives you 1 rupee. But if you get tail, you lose 1 rupee. Okay. Furthermore, if you reach 100 rupees, then casino throws you out, or if you lose all the money. Okay, that's the rule of the game. Toss a coin, it's a fair coin. Heads and tails are equally probable. Okay, you get a head, you gain 1, you get a tail, you lose 1. And let's say each of us have started with 40 rupees. So take 2 minutes. Is the question clear to everyone, first of all? Any doubts? Okay, so let's take 2 minutes and think about going to this casino and having a little bit of fun. And tell me, what would happen? What do you think is our fate, eventual fate? Can we leave the casino? Are we stuck? All of us are stuck or leave? In the casino forever? Maybe we all leave with 0 rupees, maybe with 100 rupees, or some leave with 0, some leave with 100, some remain stuck, and so on. So think about this process. Take 2 minutes, 3 minutes. Feel free to chat with your neighbors, and then we will discuss this. I'll keep quiet for 2 minutes. Okay, time up. Guys, time up. Okay, so let's see. So so what do you think? First of all, do you think we will leave the casino? How many say that we will not, we will be stuck there forever? Can you raise your hands? Okay, so let me ask you, why do you think we will not be able to leave the casino? Whoever said, you said that we will not be able to leave? Yeah.
I didn't say that we will not.
Oh, okay, sorry. So you said, yeah, why do you think we will not be able to leave the casino? Yeah. So this is the answer I always get. Okay, but that's not correct. So then what you are saying is that if I have 40 rupees, I have to get head 60 times. Is that what you're saying? Yeah, but so this is a sort of a very dramatic demo. So this is what I sometimes do. So I'm starting here. Okay, so this is my 40 rupees. Whenever I get a head, I go this side. If I get tail, I go this side. Okay. So sometimes one thinks, actually, when I thought about this problem, that's what I thought. I thought it's 40 rupees. It's a fair coin. Sometimes I get a head, maybe another head. It's a fair coin, tail, maybe tail. But what I want to do is to go to 0 rupees or 100 rupees. So think of these as those walls. Okay. So that's a 0 rupees wall or 100 rupee wall, and that's a 0 rupee wall. And once I got there, once I touch that, I'm stuck there forever. That's like leaving the casino. So let's think about it once again. So I have 40 rupees here. I go sometimes here, okay, and sometimes here. So, naively, I would expect that's a fair coin. I probably get equal number of heads and tails. So most likely, I will just sort of hang around here, right? But what's stopping me to go here? Like this, maybe sometimes more heads. And then after this, jiggling, I get here. The key point here is that there is a path, at least lots of paths, okay, that will take you from 40 to 0 or 100. And once you touch these walls, you're done. Okay, so the key thing is that there are several paths that you can take to go from 40 to 100 or 0, and therefore we will definitely get out. We will definitely leave the casino. So the intuitive understanding that we had that we will jiggle here, right? Okay, so that is not correct. So that's our deterministic thinking. Okay, so we think fair coin, but here and there. So, but actually, once you get there, you get stuck. These are called absorbing walls. They absorb you, right? Okay, so let's get back. Let's see if you understand. Suppose instead of fair coin, I had unfair coin. That means it has a chance of getting more tails. Do you think we'll still get out of the casino? Anybody who says no? Oh, good. So we've understood something. Okay, yeah, we will definitely get out, because even if it's an unfair coin, eventually I will hit one of the walls. Okay, and therefore we will definitely get out. Okay, so that's the key thing, and we will encounter this in a slightly different version of it when we come to evolution. But it's a very useful thing, this tiny little model thinking to keep in your mind when we come to genetic drift. Okay, okay, good. So therefore, now let me ask you, yeah. Yes, that's what I was going to ask you. So therefore the answer, yeah. So first of all, does the outcome depend on that? Suppose I start with 20 rupees. Maybe I start over here. Will I hit the walls eventually? Yeah. So however, what you're asking is not the question whether I'll get out. The question really is, what's the chance that I will hit that wall if I started with 40 rupees? Okay. You will hit one of the walls for sure. So we will ask this question now. What's the probability that starting with I rupees, you leave with 100 rupees? You're sure to hit one of them, no? But what's the chance now? Can you say? Suppose I asked you, I said that you start with 50 rupees. What's the chance that you will leave with 100 rupees? 1 by 2? Yeah, right? Symmetry. Right? So it's very simple to say. But what about 40 rupees? Suppose I started with 95 rupees. What's the chance that I will reach 100? It's a higher probability. You expect more, right? Okay. So that what we're just discussing right now, that we will leave, if you have an unfair coin, if I start with, I rupees 95 or 5, even if I have 5 rupees, there's a chance that I will get 100 rupees, right? So this qualitative thinking. So what you are telling me right now are your qualitative expectations. So what we discussed here are qualitative. Okay. So that's very good because at least it gets you started in the problem. It's very important. It's the first starting point. However, it does not tell me, well, if I start with I rupees, I got some probability. If I make it double, would my chance also double? Or would it quadruple? Right? In other words, it's a linear relationship or a quadratic relationship. That I cannot tell qualitatively. I just know that it will increase. Right? And that's why you need to have some models, some calculations, some simulations. So therefore, I would like to stress this. So the whole point about quantitative biology is to get these numbers. Of course, we measure numbers in simulations and experiments, so on. But this sort of quantitative thinking, it is pretty hard, especially if you're coming from experimental side, because you just don't have enough data sometimes, many times actually, right? Okay. So first question was, what's the probability that starting with some I rupees, you hit one of the what are called absorbing states? Don't worry too much if you are not comfortable with those words. But if you look up any book, any of those books, you will find this word. So better get used to it. Okay. So that's one typical question. Yes, please. Can you please use the mic? I can't hear you.
Sorry. So here we are dealing with the finite case. If we deal with the large numbers, are we still going to hit the edge? So here the starting value was 40 and extremes are 0 and 100. Instead, if the starting value is 4 lakh and the extremes are 0 and 1 million.
Starting is what?
4 lakhs. We are dealing here with the large numbers, 4 lakhs.
4 lakh.
Yeah.
But you are, so you are, then you cannot enter the casino, no? It is checking you out anywhere at 100.
Are we still going to get out of the casino?
You are supposed to enter the casino between a value.
Okay.
Yeah, I should have clarified that.
It's a constraint.
Huh? Yeah. Oh, the casino. Yeah. Instead of 100, you have 4 lakh. And you started with 40 rupees. Very good. Will I get out of the casino? This wall is far off. Yeah, very good. That's my next question. So how much time does it take? Okay. Starting with I rupees, I leave with something. And now he has brought up one more parameter. So this is this object. I leave with, so that means, let me just rewrite this. I start with this I and leave with the n rupees. Okay. And how much time does it take for this to happen? So these are the two typical questions that we ask when we are dealing with problems in evolution also, and I'll describe them again. These are called probabilities of fixation and time to fixation. And I'll review that again. Because, yes. Yeah. Yes, that's right. Very good. Thanks. And point is fair or unfair. This is going to translate into whether you have selection or not. Okay, when I come to about evolution. Okay, good. Yeah. Okay. So these are the questions that we ask. And there's a sophisticated theory, which I'll mainly mention because it's just too much math. And it's a part of what is called theory of stochastic processes. Okay. And there are several ways we can do this. Again, if you look up these books, in population genetics literature, it's called diffusion theory. But if you're a physics person, this is called Fokker-Planck equation. Okay, we will not worry about what these equations are and so on. I will, if there's time, I will just merely show them to you at some point. Okay, but we will not worry about that. So I will just give you some formulas and all, and we'll just merely analyze them. Okay. Okay, so now that we know this qualitative picture and we have some idea about maybe some quantitative thing can be done, but we will not do that today. I will now jump to what you are here for, namely evolution. So I'll come back to that. And I will define a simple model. Again, it's a very standard paradigm, very standard model in the subject. So I'll show it and describe that to you. And I want you to think what you think is going to happen. So this model, okay, right now, let's not even give the name. Okay, so I'm thinking of a population which has just 6 individuals. And there are 2 alleles. A1 and A2. And these are neutral. There's no selection. They're neutral. They have equal fitness. So A1 and A2, they have equal fitness. I've just stuck a label to them. Okay, just to distinguish between them. So these are neutral. And there are no mutations. That's very important. So you cannot have A1 become A2 and A2 become A1. Why am I doing this? Of course, in real problem, there's everything. It's the standard approach that, is very complicated. So let me just break it down in tiny bits, digest that, and then I'll put the other things together. So right now, we're just going to focus only on stochasticity. Okay, so that's the sort of philosophy here. Okay, so suppose I have these 3 A1s and 3 A2s to start with. Okay, let's say they are bacteria, so that they replicate. They make, each of them makes their 2 copies. Okay. But this petri dish has food only for 6. In other words, every system, every population has a finite carrying capacity. It has food only for 6, and there are 12 of them. So I need to kill half of them. So irrespective of what their label is, because they have equal fit, I will just pick 6 out of them. That's called random sampling. Irrespective of their labels. The word that you will see in the books. Okay, so I picked 6 of them. Maybe this dead, this dead. Say. Okay, so then I form the offspring generation in which I have this A1. Okay. So maybe both the offspring of this one lived, and let's kill both of them. And let's say these 2 stayed and this. Okay, So starting with equal number of them, everything goes very, unbiased because they have equal fitness and, very democratic, equal number of them, equal fitness. But because of the sampling, just because I picked them, okay, there's a chance that I wind up getting 2 A1s and 4 A2. Do you think it's possible I get all A2s? Yeah, sure. Yeah, all A1s is also possible, right? Can I get 3 A1s and 3 A2s? Yeah, good. You're making progress here. Good. So then this is your process in which you have reproduction and then you have this random sampling, and we can now think about what will happen to this process. So again, take 1 minute this time and think about, if I run this model in a computer or in my head, what do you think is going to happen? Yeah. Yeah, very good. Okay, so what he's saying is that, you have now 4 A2s, they will become 12, but there will be 8 of A2s. So there's a more chance of getting A2s out, right? And therefore, everybody will become A2. But is that the only answer? Everything can become A1 as well. So that you said is correct. It's a possibility. Okay, because of the reason you gave, that I have 8 of A2s, but it's possible that I get next generation, all 4 A1s and 2 A2s, right? And therefore, maybe A1 wins. This is same as what we just did for the gambler's ruin problem, in which you get out. That means you get all zeros or all hundreds, right? Okay. Similarly, here, you get eventually all A1s or all A2s. Suppose I get all A1s, then I say that the A1 allele has fixed in the population. That's the eventual fate of this population. Okay. So if I ran this program again, sometimes I'll get all A2s, sometimes I'll get all A1s and so on. Okay. So if I get all A1s, then I'll say A1 has fixed and A2 has been lost. And the questions that we're asking are, what's the chance that A1 fixes? If I started with, here I started with 3 copies. I can start with 1 copy, 2 or 5 copies, right? Just like I said, I rupees. Okay. So therefore, when asked what's the probability that A1 eventually fixes, starting with I copies. So if you like, maybe A1 is a mutant. So when you're just starting, when the coronavirus just came, I mean, one of those mutants, right? When it just comes, it's present in very small number of copies, right? Okay. And this question is very relevant because it's asking, when it just came in small numbers, right? Will it spread? That's what fixation is telling you. What's the chance that it will spread? What's the time it will take for it to spread? So that's called fixation time. Okay? So we will not worry too much. I will just give you formulas at some point. Is it much okay with everyone? Okay. Okay. So now, okay, so that we have discussed, but I want to still answer this question of fixation probability, not using a huge amount of math, but drawing what are called genealogical trees for you, so that you learn something more, just a bit more. Okay, so let me just draw this for you, and then I'll explain. Everybody can see on this side? So I'm just drawing, say I have 5 individuals. In the next generation. Okay, so see this model I drew, I made this model for you, right? You can simplify your work by saying that it's like you can remove the in between step. It's good for us to think what's the biology, but if you want to simulate this model, you can just forget the in between step and say that this one chose this parent with a probability 1 by 6, 1 by 6, 1 by 6. Okay, and this is called the neutral Wright-Fisher model. It's very easy to simulate, and some of you expressed that they're comfortable with math and simulations. It will be nice if some of you can simulate and share the trajectories and see how it looks. Okay, in Slack you can do that. You can post it if you have some answer. Okay, so let me explain that. So I'm going to intermediate step of doubling and removing and all. I'm going to just skip that step. But you can think in your head. Suppose I have this population to start with, and let's say I got this. Let me just write it and explain. So okay. So what I've drawn for you here are that we started with 3 A1s and 2 A2s. Maybe in the next generation we had 2 A1s. The parent of this guy is this, and maybe this one had 2 individuals and this one had none, right? So what I'm drawing for you is this genealogical tree or coalescent tree. We will not bother too much about it, but it's useful to know. Okay, so let me just show it again. Okay, so you can sort of trace it back here. Something is not right here, yeah. Sorry. Okay. So the reason I want to draw, and I think I probably messed it up a little bit, is that if you eventually, as you said, suppose all become A2, and you draw these trees, that means you ask who is the parent, and you draw these links, that you will find that all of them trace back to the same ancestor. So in this case, it happens that this is the ancestor of them, right? So that's sort of obvious also, right? Okay. So the question that I'm going to ask you is, suppose all are A2s, okay, and these A2s trace themselves to this A2. Okay, they could have traced back to this A2. If they were all A1s, they could have traced back to any of these A1s. So all the initial ones are equally likely to be the ancestors. Is that much obvious? Yeah. So yeah, I think I just messed it up the links. So I'll draw it again later. I'll post it in Slack. Okay. Yeah.
So like there are 5 at the end, where 3 of them can be from one A2 ancestor and 2 of them can
No, at some time it will happen, but eventually, if I wait long enough, they all will trace to the same ancestor eventually. Yeah. So in between, yes, you're right. Right. So these 2 came from this guy, and this one came from this. But eventually, if I wait long enough and I had drawn this correctly, they all will trace back to the same ancestor. Okay. So that means these lineages are coalescing to the same guy. Okay. So now that they all ancestors are equally likely to the parent, can you think and tell me, 1 second, can you answer this question for me? If I had I A2s to start with, what's the chance that my eventual population is A2? So I have 2 A2s there. what's the chance that I will get all A2s? Think about it. In the meanwhile, I'll answer your question. Yeah.
Even if we start with an infinite initial population, infinitely large initial population, is it the case that all A2s are going to be from a single ancestor?
No. If you start with infinite, yeah, that's still true. Infinitely, first of all, realistic population is never infinite, right? So, but let's say a million, billion, whatever new large number you can think of.
Theoretically.
But that will happen. Eventually, all will go to the same ancestor. That's still true. Yeah. Question? Sorry, answer? What do you think? I by N. Yeah, very good. Very good. Because all were equally likely and have I copies. So I had 2 copies. So it could have been this one or that one. So it's enhanced by that amount. So it's just I by N. So you have calculated the fixation probability in a neutral population. That's really good. Okay. When we come to selection, it's not going to be easy. Okay. But keep this little thing in mind because it's very, very useful. Yeah. So, yeah. So I think maybe Deepa will discuss molecular clock. The rate of the molecular clock you can calculate using this result. So when she discusses that, use this and maybe we can discuss that again later. Okay. Now you brought up this discussion about infinite population. So you see this probability, suppose I started with 1 copy of A2, it's 1 by N, and can be very large. So what's the chance that A1 will fix? It's very, very small, no? So should I worry about this stochasticity? It's complicated, no? Do you think I should worry? Suppose I have 1 million individuals start with and just single copy, right? The probability that A1, that A2 is single copy and I have population of 1 million minus 1 A1s. The chance that that A2 will spread is 10 to the power minus 6. Does it matter? Should I care? Why should I care? It looks pretty, complicated to me. In other words, does random genetic drift matter? Should you, bother about this lecture? Any views? Yeah, but this is a large population. Should I still bother? Small population, you're right. If I had 10 individuals, yeah, 10% probability, right? But it's 1 million. Should I worry? So if it's a very large population, typically you don't have to worry, but their population are finite. So you have to worry in the sense that answers change only a little quantitatively. So some value, something I measured, instead of 0.2, I will get 0.21. Okay. But sometimes, because the finite population, the answers can be qualitatively different. If I have N, it's 1 million, but still, you will not have polymorphism. You will not have A1 and A2. Eventually, your diversity will be lost. Okay, which is qualitatively different answer, right? So this simple thinking that we have that, there will be diversity or we will hang around in the mean and so on, that can be very different when you have stochasticity. Is this clear to everyone? This much? Okay, let's see how much time I have. Okay, I have 15 minutes. So I will explain to you one experiment. Okay, so this is an experiment to see whether or when, what is the effect of random genetic drift. Okay. So,
Ma'am?
Yeah.
So in case of population size, does
population
it
size.
also depend on individual size?
No. The fixation probability.
Like there are 1,000 ants here, and if I keep my leg on it, so most of them may die. And compared to, let's say, some large insects, like
Yeah.
very few of them will die. So
That's not random sampling that you're killing them. I mean,
no, I'm just walking,
Hmm.
and if I keep
oh, by chance you
chance.
happen to. Yeah. Okay. So in that sense, you're saying that your leg size has some size and individual size. But I think then that's more perhaps bringing in some kind of selection also, no? Because you are the environment. And that's the little fly versus a big elephant. I don't know if that, I'm a theoretician, remember? So that's what I would think right now. Okay. But yeah, please feel free to think, I mean, ask questions, even if I can't answer them satisfactorily all the time. Okay, so I want to explain this experiment. So this is a massive experiment, very impressive. So by this guy called Buri in 1956. Okay, so he's dealing with Drosophila melanogaster, which is a diploid sexual population. So what he had is these flies which have this mutation BW75 and BW that influence the eye color. So when he has this homozygote, the eye color of the fly is red. That's the wild type. For the homozygotes, they had the eye color white. So this is the phenotype. These are the genotypes, and those are the phenotype, eye color. Okay, and then we have the heterozygote, which had eye color orange. Okay, so he started this population of homozygotes. Sorry, heterozygotes with 8 male and 8 females. Okay, so here just very small population, 8 males and 8 females, and a Drosophila, as far as I know, and he puts them in some test tube, and the mate fly, produces about 100 eggs or so. So you have 100 juveniles, and then irrespective of the color of the eye, he picked out 8 males and 8 females. Okay, so that's the experiment. So you start with this, and then the mate, okay, juveniles, and then irrespective of the eye color, that means randomly sample eight males and 8 females. And this is a really huge experiment. He did it for 19 generations and 212 such populations. 212 replicates. Okay, and then he recorded the frequency of, this is the allele frequency is recording, which is what I told you, right? Of this BW75 in each generation. So first question that you will ask, you should have asked, is that, is there selection here, mutation here? So I'm assuming that mutation does not happen because generally the rates of mutation are low. And it was checked that there's no selection. How would you check that? So he established that first, but there's no selection for this. I will leave you guys to think about it. Okay, but it's an important question to ask. Okay, and then he has these frequencies, and then he plotted the distribution of, I don't know, BW75 allele as a function of time. So I'll draw that for you roughly, but I've posted this in Slack also where you can see the proper picture. But what I would like to do is to ask you how to understand what's going on in this experiment. So let me draw this for you first. Everybody understand what's the meaning of distribution? Okay. Some of you are very good in math, simulations, and so on. Those who are not comfortable, please go and discuss. What does it mean to make a distribution? What does it mean? How do you make the histograms? If you've forgotten, there's no shame, there's no this thing, just go and learn. Okay, so this is what he finds. First of all, I want to know, suppose in generation 0, the number of BW75 alleles. How does it look? How many BW75 alleles I have to begin with? I'm starting with heterozygotes, 8 male, 8 female, 16. That's correct. Can you explain? It's a heterozygous, one BW, one is BW75, right? 16 individuals, 32, and because it's heterozygous, so 16. Good. So initially at 16, we have here 212. Right? Okay. What do you think will happen if I run this experiment? So we will fill in the bits in between. That's again a very useful thing to do, to take limits. Generation 0, I know how to plot this. I know something, what happens if I ran this experiment instead of 90 generations, if I ran it for 190 generations. Can you tell me what do you think this distribution will look like? In between things will fill. Flat. Okay. So option 1, it looks flat. Any other options? Yeah, it's the number of PW75 alleles in the population as a function. Oops, sorry. Yeah, as a number of the number of populations. Number of PW75. Sorry, genes, and it's the number of populations. Thanks. Yeah. So this is the number of PW75 alleles. And when I started the experiment, all 212 populations had that, right? That everybody agrees, because that's my initial condition. I ran this experiment for 190 generations. What do you think it looks like, this distribution? You know the answer, that's why I'm asking. Yeah, please. Normal. Okay. Yes. Can you speak louder? Yeah. Yeah. So here is another option. Yeah. Any other option? Nobody likes things like, no? That's it. What's Q distribution?
Is maximum towards one end.
Okay. So something like this, but more.
Continue.
Okay, I see. I see. So like this, something like that. Maybe something like this. I don't know. Okay, so we have enough options now, and the winner is Y. So now it's time for us to synthesize, to think what we learned about Gamler's ruin, what we learned about Wright-Fisher. This is the answer. Can you justify that for me? So he already said, but can you relate to why that's the correct answer? What will happen eventually? There's no mutation, right? Wild type and mutant cannot become each other. Just by this drift, he's sampling them, right? He's collecting out of those 800 juveniles. He's just randomly selecting. So it so happened in the Wright-Fisher thing that I was drawing for you. Eventually, all of them become BW75, or all of them become BW type. It's exactly that funda. And that's very nice because, it's some sort of at least qualitative check of the theory. So in between, how you go from here to here. So it's best if you look in Slack. I posted that little diagram of his data. Okay, and then you can see how it's evolving. Okay. Okay. Yeah. So this is at 190. But in between, of course, the guided experiment. So you have answers in between generations as well, which is in Slack. Yeah? No, how can it be continuous? It's just 0 to 32. 0, 1, 2, 3, 4, 32. It's not continuous. Okay? Okay. How much time do I have? Okay. So, yeah. So I just want to do, I think I'll just make 2 points before we break. So for the mathematician physicist types, I said to you very briefly, I said that you can do this theory. This distribution that he has got, actually you can calculate it. You can get the entire distribution. It's not enough to just say how much probability and how much time. You can find the whole distribution. That's a lot of information. Okay, and I'll just mention it to you, because some of you have seen such things. So one can write down an equation for the distribution. Probability that the frequency is X at time T. So one can write down a partial differential equation for it. Don't worry too much. Those who know, let me just address this to them. Okay. This is the equation that you get. Some of you have seen such an equation without this stuff. Okay, that's called diffusion equation. Okay, physics especially. The physics problems that you do in your undergraduate and so on, that's different because of this factor, which is coming actually due to heredity. These problems, although we are using similar approaches and so on, the equations and the model that we deal with are different. All the phenomena are similar, but the answers and all are different. Okay, so that's one comment I wanted to make. One can find, using this kind of theories, something called heterozygosity. We are dealing with diploids. You can find out the heterozygosity, which is a measure of diversity in the population. Initially, the population is very diverse, right? And later on, at the end, 190 generations or so, there's no diversity. Everything has become either this type or that type. There's no heterozygosity. Everything is all are homozygotes, right? Correct? So how does this diversity decay with time? So we don't have time, unfortunately, to find it. But using this kind of stuff and other simple stuff, one can find the answer to that. And that's what he compared with his experimental answer. So I'll just plot that for you. So if you measure heterozygosity, by the way, one question. I said, okay, I can plot BW75, etc., etc. How do I know? There's no genetic sequencing or something, no? It's 1950s. How do I know that I have? Yes. Okay. Yeah. Because this is a simple trait, right? You should try to think about how is he finding the number of alleles. So I think his data is there in the paper. Try to see, it's always good to go back and just break it apart and go into any detail and work it out. So if you look at his data and see if you can plot the distribution that he has, it's a very good exercise to do. Okay, so heterozygosity, it was 1 initially because all were heterozygotes and decreases towards 0 pretty quickly. I'll give you the formula. It decreases exponentially. And the time scale depends upon the population size. If the largest population, it takes longer. Right? We have qualitative expectation anyway. If you have large population, it takes longer to hit one of these walls. Right? So it goes like that. And that's what he measured and compared that formula with the experimental data. He got a very good fit, but not with n equal to 16. He had to use n equal to 10. Then he got a good fit. So why? Well, because actual things are very complex. These are simple models. Although they do give some insight, there are other stuff going on. So for example, here's something assumed, which is called random mating. That all these 100 juveniles, this guy and this guy, they randomly mated, but maybe they have a preference. I have not taken care of that, right? There could be other factors. So all that complexity can, at least in this case, could be subsumed in a single parameter, which sometimes you will see in books, called effective population size. So for this experiment is 10. It's not always possible to do that. In fact, nowadays we find that many problems that we do, more detailed ones, it's simply not possible. But many times, sometimes it's possible, and therefore it simplifies things. Okay, so that's effective population size. And one more really last comment. We can also calculate using these kind of theories the time. I said 190 generations. but we can calculate that. And it turns out to be 2.8, the mean time, 2.8 times n. So the time to reach here so I don't have a simple argument for it, but it turns out to 2.8 NE, NE is 10, so 28 generations. So you expect on an average, you would reach here 28. He stopped at 19. And if you look in SLAP, you will see that actually he did not reach this. So 212 by 2, each of them should be right. But he didn't reach there because he stopped at experiment 19. So theory is very useful because at least it gives you an idea. Do I have to apply for a grant which will allow me to go 190 generations or another 10 will do? So at least it gives you some pointers. Okay. Okay, so I think I'm done. Yeah, okay, I'll stop here unless there are questions. Yeah. No, it's just a fit. Because here's the formula. If he puts n to be 16, he doesn't get a good fit. He changes, plays around, 16. Instead of 16, you put 10, works great. So that's my answer, right? But why it is ineffective, can we understand from the details and all? As far as I know, that's not known for this experiment. But it's a useful concept. Yeah? Selection is very different thing. So today we assume that all are equal. Okay, it's just a label which is neutral. Okay, so we'll start with selection next class. Yeah.
Can the n is effective population size?
Yeah.
In like when you're fitting a model, it's possible for it to be higher than that.
Typically it's lower. Like for example, humans. I mean, I think it's always lower. For example, humans, our census population size is what is it now? 7 point something billion, right? But our effective population size is some 40,000. Yeah. Yeah, but I mean, so it's a measure of the diversity in the population. So, it's just that.
But if you were to let's say, constraint mating, instead of random mating, if you were to constraint mating that it is uniform.
Yeah, so then you have to change. Yeah, then you can actually find an effective. You can build in models, not for Drosophila maybe, but things like if there's inbreeding, for example, you can put in some factors. There are detailed models which will tell you how much is ineffective depending on those inbreeding coefficient. Suppose you have sexes need not be equal. Then how much does it change? So those simple things one can do. So it's not just an arbitrary fit. One can do a bit more. But here I've assumed all those things to be irrelevant to this experiment. Anything else? Okay, thank you very much.